Right. So thank you for the invite. So um as Daniel said, I'm Nick Rawlence. I run the Targo Paleogenetics lab, and what we basically do is use paleontology, archaeology, and ancient DNA to reconstruct prehistoric ecosystems. So whether it's the time of human arrival or back tens of millions of years, look at how our dynamic geological, climatic, and human history have influenced those ecosystems, and then through conservation paleontology, how can we use that moving forward for conservation management? So the title from Brobdingnagians to Lilliputians, I've stolen from Gulliver's Travels, where Gulliver went around the world um, and discovered giants, the Brobdingnagians, or Lilliputians, the little people, or dwarfs. And so this talk really shows how we can use these paleo tools to study not just the giants, but some of our smallest animals in the ecosystem. So, um, as with any talk, I'm just the balding messenger, and a lot of this work has all been done by my fantastic bunch of postdocs and postgraduate students, but also wouldn't be possible without the support of museums around the country, big and small, funding bodies, but also iwi partners. And I've put some of them up there, but by no means all. So basically, all of this should be fairly familiar, but we've had a very dynamic geological history. So around about 82 million years ago, New Zealand decided it wasn't happy in its marriage with Australia, so it decided to divorce and start drifting through plate tectonics out into the Pacific Ocean. And I grew up with, certainly in primary school and high school, that dispersal wasn't really a thing, and that the prevailing view, personified by David Bellamy, was that the flora and fauna of New Zealand was all once part of Gondwana. So I grew up knowing this was all, thinking that all of the animals in New Zealand were due to Gondwanan vicariance. So that they'd stayed in New Zealand, and New Zealand passively drifted across the ocean through plate tectonics, and they were having Devonshire tea. So much so that I remember reading New Zealand Geographic, and I've still got this issue, where it is basically showing a suite of our fauna and suggesting they're all Gondwanan. And for some of them, we know that they are most definitely not Gondwanan anymore, like your Lancewoods, your Rata, your Takahe, for example. And but well before that, Charles Fleming, who, if Ewan Fordyce is the father or grandfather of New Zealand paleontology, we can think of Charles Fleming as the great grandfather of New Zealand paleontology. In his seminal publication in the 60s, was suggesting, just based off our fossil record and the distribution of plants and animals, that there was dispersal and vicariance. So we had this endemic archaic group containing, we've got Leopelma, Moa, Peripatus, Tuatara, Kiwi, flax, and mystacynid bats that had always been in New Zealand. Some of those have held up and some haven't, but we've actually got all of this dispersalist group coming from different areas of the world. And this was long before the days of genetics. So all from a paleontological and biological perspective. But since David Bellamy's everything was Gondwanan, we've kind of gone on these swings and roundabouts and these pendulums. So as New Zealand drifted away from Australia, as Zealandia drifted away from Australia, the crust then became less buoyant. And so it started sinking beneath the waves. And it's been quite controversial. I remember this cartoon coming out about the time Hamish Campbell published his book In Search of Ancient New Zealand, suggesting that all of New Zealand drowned, and Tuatara had basically swum to New Zealand. So the cartoon says like only another 100,000 years of paddling to go. So the debate has raged about whether 80 to 90% of New Zealand went under the waves or not. We know from the genetic record that you can look at some of our iconic New Zealand animals like wrens, and we can see population bottlenecks. And from the fossil record, we can see genetic faunal turnover or biological turnover. And it got to the point with like Hamish Campbell suggesting there was all this drowning, and Mike Pohl, who's a geologist based down here in Otago, who was suggesting that the present New Zealand vegetation couldn't be called Gondwanic, either in character or in the sense that it evolved in isolation from pre-Gondwanan lineages. But we know from all the fabulous work of Daphne Lee and her group that there was land at the height of the drone. We have Folden Maar, which needs no introduction, being this Maar deposit out near Middlemarch that contains this really rich terrestrial and aquatic record preserved or basically pickled in this lake. That's vegetation and climate is similar to southeastern Queensland, and has some of the oldest fossils we have in New Zealand, like of our galaxids or ants or spiders, and leaves with stomata, the breathing parts of leaves that take in CO2 and breathe out oxygen, still preserved. And we have a few million years later, St. Bathans. I'm just going to minimize floating meeting controls. And we have St. Bathans, which is one fossil outcrop from this giant Lake Manahirikea, which is about 5 times the size of Lake Taupo, that I've just managed to spend 11 lovely days ignoring my emails at digging up fossils with a team we've got with to Alan Tennyson to Papa, but also Lizzie Steele from Cambridge University. So it's a lacustrine lake deposit in central Otago, and this lake basically stretched east to west from Bannockburn to St. Bathans, and north to south from the Waitaki River Valley to the southern Nevis Valley, which is about on par with Kingston at the bottom of Lake Wakatipu. And the dating's a bit controversial. It's oft quoted that it's 16 to 19 million years old. Pollen evidence from Mike Pohl's research is suggesting that it's probably 12 to 13 million. And then recent research published out of the Department of Geology down here at Otago is suggesting it's no younger than about 10 to 11 million years based on dating of lavas in the Maniatoto underlying St. Bathans sediments. And the deposit contains many of the ancestors that we would recognize as New Zealand. And so you've got ancestors of Kiwi that could probably fly. You've got tuatara, mystaciniid bats, our skinks, geckos, and frogs. But it also contains these fantastical animals. I like to think it's like the Miocene version of Alice in Wonderland. There were paleolodids, which are relatives of flamingos. You've got giant land crocodiles, giant turtles, these weird birds no one knows what they're related to, relatives of the Nicobar pigeon as well. And if we fast forward through from this Miocene, I suppose, hothouse, we get to the Pleistocene ice ages. So if you've ever watched Game of Thrones, it's the White Walkers cometh. And so you've got from about 2.58 million years ago, these repeated cold glacial, warm interglacial cycles, around about 19 to 20 of them, where you go from this completely alien world of the last glacial maximum, where your forest retreats to the northern areas of both main islands. You could walk from Auckland to Dunedin. You don't need to take the ferry anymore. And most of the South Island is all under ice. So this purple and light purple is barren ipon tundra. And you've got this open grassland shrubland mosaic. Flipping to the Holocene, what we're kind of familiar with now at the time of human settlement, New Zealand cloaked in forest. And on top of that, very recently, we have the arrival of humans. So they had successive waves of humans from Polynesia and Europe, this devastating impact on New Zealand's wildlife through overhunting, habitat destruction, but also predation from a whole horde of furry predators that has caused this massive amounts of extinction. So we've lost probably 80 species of birds and counting since the arrival of humans, which represents up to about 70, 80 million years of evolutionary history. So if we got rid of humans and let the clock play out, it'd take about 70, 80 million years to be able to evolve the sort of diversity that we had. And so that's really the backdrop that we work in terms of reconstructing these ecosystems and how these forces have impacted on these ecosystems. So to take you through, I suppose, kind of like geological time. This is work that we're doing with Alan Tennyson from Te Papa and Lizzie Steele at Cambridge University, where we're going to St. Bathans and we're doing excavations. We're bringing along all of my postgrads and we send out emails to the geology department, who we work closely with, and around the university, who actually wants to come and get hands-on paleo experience. And so one of the what we discovered, this was at Marta 10, not Marta 10, sorry, Marta 9 at St. Bathans is on the left. We've actually got this fairly big chonky bone. So this is the holotite that had been in Te Papa for years, but wasn't really recognized as anything different. And we started finding very similar large chonky bones in Marta 9. And so we compared them to the bone on the right. So that's Mytodorna, so Mycene todorna. It's not an ancestor of paradise shell ducks, which only turned up in New Zealand about 1.5 million years ago. We're talking kind of like a cousin in the same family. And so when you do that comparison, what we can see is that Mytodorna catriona, that we ended up naming it after my mum, who inspired my love of natural history and immortalizing my kid's grandmother as a giant fossil duck, much to the amusement of our family. It falls right outside the range of the rest of the St. Bathans natted. So it's not sexual dimorphism like what you see between paradise shell ducks, males and females, and we've got very different proportions as well. So we're talking about a shell duck that's probably about 30% bigger than the current paradise shell duck and quite chonky. So it's fitting in with that familiar yet fantastical world that you find at St. Bathans. But what we also have at St. Bathans is two different species of grayling. So the extinct New Zealand grayling, which went extinct about the 1920s, is kind of like the grey ghost of the fish world. It needed pristine, wide, navigable rivers, and it would appear at night in these shoals and just as quietly disappear. And so there's been this big debate around, is the extinct New Zealand grayling the most closest living relative of the Australian grayling? And when did it arrive in New Zealand? And we have fossils from St. Bathans, and there are some fossils from the late Pliocene as well. So we wanted to try and work out what it was related to. And all of these grayling are in museum specimens, and they're all formalin fixed, which isn't great for ancient DNA. And so one of my student extraordinaires, Lockie Scarsbrook, who's currently at the University of Oxford doing paleontology and ancient DNA PhD there, has got the mitochondrial genome. So this is the DNA that's passed down through the female line from mother to daughter from the extinct New Zealand grayling. And we reconstructed this evolutionary tree, or whakapapa. Think of it as a family tree. And this is Prototroctes oxyrhynchus, comes out as most closest living relative of the Australian grayling, Prototroctes marinara. That's all well and good. They're closely related. The divergence is quite deep, so they're not the same species, which is good. But we want to know when. We can't use St. Bathans fossils as calibration points to anchor divergences in time because, as I was suggesting, we don't actually know the date of St. Bathans. It's not as tight as 16 to 19 million years. It's probably anywhere between 10 and 20 million years. So we used some calibration points in Osmeridae. This is your northern hemisphere radiation of uh, fish, and you've got your southern hemisphere radiation, your Retropinids, so your smelts and your graylings, and to calculate an evolutionary rate, so how much change do we see over time, and we've got a divergence date down here of no more than 23 million years ago, which fits with the St. Bathans grayling, the two of them being the ancestors of the New Zealand one. But it's really interesting because the date we've got here of about 23 million years, being the earliest time they could turn up in New Zealand, is really similar to the molecular dates that geneticists are getting for the arrival of galaxids in New Zealand. And that's gelling also with the fossil galaxids that Daphne Lee is finding in Folder Mar. So it's suggesting that while we had land through the Oligocene drowning, we may not have had many freshwater habitats because of very low-lying relief, and that you may have had this biological turnover across the drowning of freshwater fauna that only managed to recolonize after the drowning event as you got uplift of the land relief and rivers and lakes beginning to form. So we're managing to use paleontology to help inform, ancient DNA to help inform paleontology. So if we fast forward a bit out of this Miocene world into the Pliocene, we're about 2.5 to 5 million years ago. We're beginning to cool down, but we're still quite wet. And this was a discovery made by Al Mannering and Bruce Marshall. And it sat into the bowels of Te Papa for ages until one of my students, Luke Easton, had a look at it when he was doing looking at frog bone shape. And he discovered these fossil bones. So we've got A is a modern fossil, B, C, E, and F are this frog I'm about to describe. G is another frog, and then D is one of the Miocene frogs out of St. Bathans. As he started looking at all these bones, and whereas we've actually got something new, and we started looking at all the osteological characters, so your bumps on bones, measurements describe the shape, and what we've got is this completely distinct frog from the Pliocene about 3.2 million years ago, and it's kind of like the grandfather of all of the late Holocene, Pleistocene frogs that we have in New Zealand. We named it after the late Phil Bishop, who was a professor in our department that passed away a few years ago from cancer. But the site's really rich, and we're going back because the bone in G is not our new frog, Bishop's frog. It's actually something completely different. So for this tiny site to have two different species of frog potentially is actually really interesting. And suggests quite a rich diversity. But with the other interesting thing is you wouldn't think of North Canterbury up the Waipara River now as a really good frog habitat. It's barren, it's dry, it gets really hot. But 2.5 to 5 million years ago, it was still wet and forested. So you've got this quite cool keystone climatic fossil indicator. So around 2.5 million years ago, suddenly everything changes. Most of New Zealand's geological history, we've been completely forested. We're seeing forest birds turn up throughout the geological record, or the tupuna ancestors of forest birds alive today. But 2.5 million years ago, New Zealand starts going to the glacial period. Our habitat starts looking a lot like Australia. Wide, open, there's a whole lot of niches or job vacancies open up. And we start seeing the arrival of the ancestors of our quintessentially New Zealand birds. So like little eagle turns up and evolves into Haast eagle. The Australian raven turns up. The Australian coot. The Australasian harrier evolves into Isles harrier. Purple swamp hen pukeko turns up and evolves into Takahe and Moho, the North Island. And we're getting this massive influx of generalists or open habitat specialists that arrive here really rapidly over a few million years, evolved into these quintessentially New Zealand things. So the little eagle is the smallest eagle in the world, evolves into one of the largest eagles in the world, Haast eagle. Now, some species go extinct after human arrival and are never replaced. So Haast eagles never replaced the raven, the coot. Other species go extinct. And with the habitat clearance from Polynesians and Europeans, we're kind of history repeats itself. And we're creating the conditions and the job vacancies like you had in the Pleistocene. And so we get, for example, swamp harrier around everywhere today, only tuned up and within the past 400-500 years. Pukeko is the same, as only tuned up since the extinction of the North Island take and the near extinction of the South. And with forest clearance, you get all of this changes in drainage and development of wetlands. So we're getting other wetland birds like ducks, the bitterns, the herons, the woodsaws, and the grebes all suddenly start arriving. And so work by my PhD student, Pascal Lubber, has extended all of this stuff and is showing that we're getting this big, massive influx in faunal turnover event when the ice ages start. So that's kind of how glaciation can help, but glaciers have traditionally been thought of as really destructive forces for biodiversity. You think of the giant ice sheets in the northern hemisphere, and that when the ice sheets come in, all of your animals at higher latitudes retreat to like utopia refugia areas in like the south of Portugal, France, Spain, Italy, Greece, such hardship. Go through bottlenecks, and when the ice retreats, they expand back out. And so you get lots of genetic diversity down in the refugia and not much genetic diversity up in previously glaciated areas. And so you can kind of think of ice sheets destroying biodiversity. Yet a lot of the evidence that's coming out in with some ancient DNA stuff, so some stuff I've done on Moa, but also stuff looking at modern DNA out of rock green and some of our invertebrates and lizards, has shown that glaciation in temperate areas, so like the Himalayas, the Pyrenees, the Andes, New Zealand, can be quite a creative force. So you get these continuous populations like you got in C here, the early Pliocene. Glaciations come in, and especially with the uplift of the Alpine Fault, you get Southern Alps covered in ice, and it separates these populations into two different areas. They evolve in isolation, and you get lots more genetic diversity being created. And one of the things we've been showing is it's not as clear cut as everything in temperate ecosystems is showing this pattern. And one of the good example is if we look at eastern moa. So eastern moa is one of the most common moa in the subfossil record across the eastern and southern South Island. Yet if you look at its DNA, it has about as much genetic variation as Prince Charles and the English royal family. Very inbred. It's not like the genetic variation in the audience listening to this talk. So it's this conundrum of how can you have this really, really widespread species with absolutely no genetic variation? And if we start looking at ancient genomes that my student Alex got, and we start looking at, say, Pleistocene sites, this is Ice Age sites older than about 11,000 years, and then Holocene sites younger than 11,000 years, so the ones in red, you kind of see this explosion of diversity as you go from the blue to the pink or red, and it comes out in the star-like pattern. Now, that's really indicative of rapid dispersal or radiations out of glacial refugia, or signals of domestication. And so what we find is if we start mapping all of this onto areas where there's where we think the putrid refugia for eastern moa is, based on all of the Pleistocene glacial fossils, it's down in this purple area, southern New Zealand. We actually have more genetic diversity down in southern New Zealand and less genetic diversity outside of that refugia. So we've got this really continental signal like you see with ice sheets. And this is to do with the moa tracking the distribution in their preferred habitat, in this case, forest. And kind of reminds me of this Bill Bailey quote. He gave it a comedy thing in Dunedin where he was standing, it was one of his songs he was singing, he was standing at this cross intersection with a lonely street light and only a Kiwi for company. In every direction he looked, it was quiet, it was dark, there was nothing happening. And the punchline was it was a Friday night in Invercargill. I'm pretty sure he probably said the same thing about Dunedin when he was in Invercargill. But these moa retreated to southern New Zealand and expanded out. And if you look at other species like heavy-footed moa, which a famous New Zealand archaeologist, Beverly McCulloch, described as a 40-gallon drum walking on little kids' gumboots. At the height of the Ice Age, that went to southern New Zealand, and another group went to northern New Zealand. And upland moa, the mountain goat of moa, went to the four corners of the compass in the South Island. So rather than having an entirely continental signal like in North America and Europe, and this entirely temperate signal of glacier-driven evolution promoting the evolution of biodiversity and genetic lineages, we're actually seeing every species as having these individualistic responses. And so you can't have one moa to guide and rule them all, or one response to guide and rule them all. So you're probably thinking, well, why aren't there any moa down on Rakiura, Stewart Island in that map? And it's been this big debate of whether there actually were moa on Stewart Island. Rakiura was attached to Mirahiku, the southern South Island, at the height of the LGM. And we know, for example, the DNA suggests kakapo have been on the island for 20,000 years, yet you don't actually find any kakapo bones on the island. It's heavily forested. It's really acidic, and bones don't preserve in the forest. You actually find very few terrestrial and no forest birds preserved in any of the subfossil deposits on Stewart Island. And so there's been this big debate about whether moa lived there naturally or whether they were killed on the mainland and transported to the island. And so this was a discovery that Doc made, and in the photos, Matt Smith, who's the Doc archaeologist of southern New Zealand, is, if anyone's been to Rakiura, behind Matt where I'm just dragging the cursor, so down that gully, that is the track down to West Ruggedy Beach on the Northwest Circuit. We're about 20 meters away from the track. And this was, it's not the best skeleton, but it's an associated skeleton. We've got femur, we've got tibia, we've got part of your pelvis here, we've got ribs, and we've got no cut marks whatsoever. And we've got this really dark organic rich layer you can see quite easily in A, very different to the surrounding sand. And we've got gizzard stones down here. So it's this moa that's died in situ, it's died where it's lay. The date we've got is just after human arrival. So what we think we've actually got here is definitive evidence of moa on Rakiura. Within a stone's throw, there are actual of this. There were chick remains that were found, and we don't think this was killed on the mainland, because you'd have to kill the moa on the mainland, put it in a waka, paddle it across Fovo Strait, round the rough side of the island, and then carry it. So from where Matt is here down to the sea in the background of this photo, it's about 500 meters upslope, and then dump the moa or not use it. So most of the work we're focused on is Brobdingnagian giants, but what about some of the other smaller species? So there's been this big debate around when did the ancestors of Takahe, so in red, and moho in green, arrive in New Zealand. And it's always been thought, like even right up until the past couple of years, that because Takahe and red and moho and green aren't each other's closest relatives in this whakapapa, and Porphyrio melanolotus, that's your pukekos between them, that it was two invasions into New Zealand. And what one of my students did is he went back through the fossil record, the South Island and the North Island. He got complete mitochondrial genomes from the Takahe and the moho. And surprisingly, that moho, if anyone knows Northland subfossil sites, this is from the top of Harangi Hill up Cape Maria Van Diemen, which is not the area you'd expect for good DNA preservation. And so what he's got is each of them are each other's closest relatives. And they split from Pukeko about 5 million years ago. So as we're coming out of that Miocene forested habitat after the mid-Miocene cooling, it was going to the Pliocene, Pleistocene. It's beginning to open up. And then they split into North and South Island species around 1.5 million years ago. So as the Manawatu Strait So this was the Pliocene Strait that had Palmerston North completely underwater. Sorry. Um, as that closed, you got dispersal of already flightless species across the land bridge. Cook Strait didn't exist at that stage. It's only about 400,000 years old. So when the Manawatu Strait was around, you had the northern North Island and the southern North Island was connected to the South Island. But this is a really good example of how changing ancient DNA technology coupled with paleontology can fundamentally change your answers, because it comes down to the old answer was due to a whole lot of error in the DNA sequencing that we can actually account for and correct in with the new sequencing technologies. But Takahe and Moho were everywhere across New Zealand, and the Moho is now extinct, and the Takahe is only found in the Murchison mountains and has been spread all around New Zealand where those blue shapes are. So how has the Ice Age actually impacted Takahe? So if we rebuild this whakapapa, what we can see down here is we've got the North Island Takahe, the Moho. There is some genetic structure in there, but it's preliminary. We think that Northland's different to Matakona, so that's in the Wairarapa. But we don't have much DNA from fossils. There's something about volcanic ash mixed with humidity of the North Island that, while you get really good preservation of fossils, look at their shape and everything, ancient DNA preservation is not that good. When you look at Takahe, the other group there, all of our Pleistocene Takahe, like Hodge Creek Cave System, His and Her Cave, Forest Hill, Honeycomb Hill, are all down the base of the tree, and we lose all of those. We don't see any of those lineages when we're looking at the Holocene. So pre-Polynesian. So we've already lost a whole lot of genetic diversity as you've gone from a cold glacial to the warm interglacial. And one of the upshots of that is it's suggesting that the areas they were in, which is Northwest Nelson, is not their ideal habitat, which actually explains why the translocation of Takahe into Kahurangi National Park in the Heaphy has been an unmitigated failure. It's not good habitat. They all left. They all went down to their preferred habitat based off the fossil record of dynamic edge habitats. And a lot of the dock modeling that's done of where they put Takahe is using their relic distribution as their ideal habitat. So it's a really good lesson that paleontology can inform conservation going forward. Like, what is the pre-human preferred habitat of these species, and is the relic habitat preferred or not? And with the arrival of Polynesians, we lose all the genetic variation. So that big straight line you can see here is indicative of a major genetic bottleneck. So you've gone again from the genetic variation, say, of the city of Dunedin to the genetic variation of the English royal family. We don't see another hit when Europeans arrive. The damage has already been done. So we've got a whole lot of these Lilliputian fauna that have this really deep evolutionary history, but we haven't actually been able to touch them, like the wrens, for example, because they're so small. If we try to take samples, we're going to destroy specimens. So we know that our A-considered wrens are the most primitive passerines or perching birds in the world. They've been in New Zealand for nearly 70 million years. And we know that two lineages, your Lyell's wren and your lineage that led to your rifleman, bushman, rock wren, stout-legged wren, two lineages got through the Oligocene drowning event. And we have some fossil species like Cooney ornis from St. Bathans. And again, it's a good lesson in paleo of we need to be really sure of the date, because the St. Bathans fossils, for example, have such a fundamental influence on how you actually date these whakapapa or phylogenies as well. So we need to be really, really sure of what is actually happening. And some species we don't actually know anything about them, like Dendroscanth, the long-billed wren. There is barely any bones of it at all. So one of our species is the bush wren. There's a North Island subspecies, Stokeseye. There's a South Island subspecies, Longapies, and there's a Stewart Island or Big South Cape subspecies, Variabilis. And so these species are really tied to forest. It's like what we find is the smaller the species, the more coupled or tight their association with their habitat is, as opposed to the larger species that can move around a bit. So if we reconstruct again the family tree, and this is work Alex did with getting mitochondrial genomes of small toe pads, trying to chop a toe pad off a tiny little bush wren, and then if it pings and goes flying onto the bench and floor and find this toe pad, is a bit hair raising. But what we can see is if we look at the North Island bush wren, so down the bottom around Hokanui, Rumataka Ranges versus the South Island complex, we get this really, really massive big split. And then within the South Island, we get a whole lot of this diversity in these deep splits. So up the top of the South Island, we can distinguish between Northwest Nelson around Karamea, the Marlborough Sounds. We can do within Canterbury. What I think this is either side of Arthur's Pass, and these animals don't actually see each other because they're stuck with forest. And at the LGM, you had a glacier going through, or now you've got a frost flat with tussock, and they don't want to cross. And then we see structure within Fiordland and Big South Cape Island. So one of the things you'll notice is you've got this initial big split down here, and then this big long branch with nothing much happening, and then boom, suddenly, simultaneously, something happens. And what could cause that sheer amount of structure? And we're talking about, say, between this Bealey and this Bealey, hundreds of mutations different, which is really pronounced. So if we date this phylogeny, we can see that the bush wren separated as a whole, separated from the closest relatives, the rock wren and stout-legged, around about 12.5 million years ago. We're getting increased land area coming out of the Oligocene drowning. We've got this hothouse. We've got lots and lots of forest. And around about 5 million years ago, you get the split between the North and South Island. Again, the Manawatu Strait. And then 800,000 years ago, give or take, is when we get this massive explosion of genetic diversity being created. And we think that's the glaciations. But more importantly, what I think we're seeing is an ecological tipping point in the fossil record. So that we have the ice ages start about 2.5 million years ago, and the fluctuation between glacial and interglacial is not that bad. You're getting glacial periods about 40,000 years in duration until we see the Milankovitch cycle, so the Earth's wobble shift, and suddenly you're getting much greater environmental fluctuation between cold glacials and warm interglacials, and your glacial periods become about 100,000 years long. So it's almost like the ecosystem can withstand a certain amount of pressure until it reaches a point, and it's like, no, bugger this, we're going to hang out in some nice forest somewhere and not deal with the cold, and you've reached this tipping point, and that's what we're seeing. So one of the current stuff that we're doing, which is we've got Daniel involved in this, and this is what's actually the Marsden I've got on paleontology with a bit of ancient DNA, is if you look at a fossil in the fossil record, and so this is the purple triangles, and you compare it to living species, say the blue and the red, and it looks different, you would hypothesize that it is a new extinct species. Much like what we've got in New Zealand, where we did the same, we found out there was an extinct swan that was different to the swan here now. But what happens if your fossils actually are the same as the living species and they've just undergone rapid morphological change? Much like what you've got with dwarf species of emu in Australia off of Kangaroo Island or King Island or Tasmania. Distinguishing between those two hypotheses can have major implications for how you infer extinction rates or biological turnover rates or speciation rates through deep time in the geological record. But to give you an example, if you found the skeleton of a Habsburg and compared it to modern humans, you would be forgiven for actually thinking it's a completely different species of extinct human, despite the fact it is an anatomically modern human that has undergone rapid morphological change because they wanted to keep it all in the family. And that to the point they couldn't actually have children. So in New Zealand, we have this major radiation of skinks and geckos that have been here for tens of millions of years, and we've had a whole lot of extinctions. And we've got an exquisite fossil record. So to give you an example, this the fossils of Duversel's gecko, all the stars across New Zealand. We think there are probably fossils in the southern South Island. We just haven't found them yet, compared to the modern distribution all on offshore islands. And whether it's across tuatara, frogs, skinks, or geckos, there are hundreds to thousands of these fossils. But they've been split into these, especially for skinks and geckos, not for frogs and tuatara, where there's a lot more bumps on bones to be able to tell what species they are, whether living or extinct. But for geckos and skinks, they've been split into these coarse quasi-taxonomic size bins. So it's been thought that you couldn't identify anything by shape, it is only size. And that's underpinned a lot of the conservation paleontology or ecosystem restoration done by DOC. But we can't look at it. So if we try to do the DNA, or even recently the morphometrics on these tiny small bones. So what we've got here is a giant moa tibia bottomy shin bone, and this little bone here is the jawbone, which is like half the size of my little fingernail. And if we try to actually sample these, we would destroy the bones and end up with very grumpy museum curators. So how do you solve this problem? So what my student Lockie Scarsbrook did is he micro CT scanned, in this case, the maxilla of one of these size classes of gecko, and used fixed landmarks and semi-landmarks to characterize basically bone shape. And he did this across representatives of all the living gecko species and compared it to fossils. And he also came up with a completely non-destructive ancient DNA extraction technique. So we can soak DNA out of these bones. We can do CT scanning pre and post. There's no damage whatsoever. You can still do 3D geometric morphometrics after soaking, and we can sequence complete ancient genomes. So what he did with the modern is he, so this is down in part C, is he looked at all the maxilla of all of the living gecko genera, and he could distinguish, as you can see, with these convex hulls and these 95% ellipses, he could distinguish every single genera of New Zealand gecko. So we've got Duversel's up here in red, we've got the Woodworthy complex down here in blue. So great. We've thrown size out with the bathwater, we can do it with shape, which is fantastic. And then what he did is he CT scanned all the gecko bones out of the size class and plugged them in. And some, like up here, E, K, D, J, and A fall within Duversel's. Yep, we think they're Duversel's. B down here falls with Woodworthier. Okay, that's all good. But then you've got a whole lot in no gecko's land or no man's land. Okay. Maybe what we've actually got is a brand new extinct species of gecko. We're not sure. You could, that's what you would hypothesize. But with these, we can get ancient DNA out. And so what Lockie did is he soaked all the DNA out. Again, there was no damage. We effectively went fishing for ancient gecko DNA. So just like you go fishing for fish, you attach a bait that is preferable to your prey species, and you go fishing. Here we've attached modern gecko DNA to find ancient gecko DNA. And we can build a whakapapa. And what we find is that these geckos in no gecko's land are actually Duversel's gecko, and we've just undergone this rapid morphological change. And that could be due to the impact of human colonization of New Zealand. You've gone through a genetic bottleneck, and you could have gone through a morphological shape bottleneck. Another option could be all of our prehistoric geckos at the time of human arrival were on the mainland, and all of our surviving populations are on offshore islands. So maybe there's morphological or shape change in geckos as they're adapting to a suboptimal habitat on an offshore island as they get trapped there after sea levels rise at the end of the last ice age. So if we reconstruct the evolutionary tree of geckos, what we can see is that the split between Duvercells on the North and South Islands about 5 million years ago, like with Manawatu Strait. So much so we named a new species and we got gifted the name Tohu gecko by Te Atiawa. So this is named after one of the Parihaka prophets, Tohu Kakahi, who was arrested by the English in Taranaki, sailed past the type locality Three Brothers to Nelson, then sailed down the eastern South Island past the prehistoric range to Dunedin, and then was forced to build things like my wife's high school, the clock tower at the university, and several other places. In the North Island, we see these old lineages on old islands like the Poor Knights Great Barrier and old rugged areas like Waitomo or Northland. And we can see a whole lot of lineages trapped on islands with Holocene sea level rise. In the South Island, we can see glacier-driven evolution splits into northern southern lineages around when the glaciations kick in. And we can see this contraction expansion in and out of refugia. So by looking at the Lilliputians using ancient DNA and paleontology, we're really telling these forgotten stories. But we can see shape variation across other bones in the skull. If you look at these pictures of this is one individual of each gecko genera, there's variation in all the other bones. So if we look at the frontal bone, which is this one here, we can do the same. I've got a student, Alex Brown, who has distinguished frontals from all the living species of gecko. And when you plug in another size class of subfossils, it looks like someone shot the picture with a shotgun. Or that the size classes are meaningless and we're getting the full gamut of, say, gecko and skink size shape distribution in the subfossil record. So we can really start reconstructing what's extinction, what's lost morphological or shape diversity in a very neglected part of our fossil record. And we can use these techniques to go to other species. We can look at New Zealand wrens, especially like Lyle's wren or Stevens Island wren. So just only found on Stevens Island historically. There are bones across the entirety of New Zealand that are probably multiple different species. So to finish up with, paleontology is not just vertebrates. We've also got mollusks. But if you look at mollusks alive today, you can look at all of these species and go, I can distinguish them because I've got the color, I've got the tissues and everything. Or I could look at these power shells and go, I can see different colors and patterning. But for those pictures on the left and the middle, if you didn't have all of the tissues or the color, you would say all three of those are the same species. And mollusks make up a lot of our fossil record. If we're thinking of upraised beach deposits around Wairarapa and Rowlington, old tsunami deposits or subfossil sand dunes of Northland, we can't get, until now, we haven't been able to get DNA out of them. So you can't really ask questions of like how many species of giant flax snail have we got? How much genetic diversity is lost, or shape diversity? What has been the impact of climate change in humans? And so one of my students, Kerry Walton, is now curator of mollusks at Te Papa, developed this method where you can extract DNA, go fishing for that DNA with 300-fold greater efficiency than any other method, and sequence ancient genomes. And he showed this with power. We've got lots of genetic diversity. We've got different groupings and new species that we're writing up. And so we're using this method to start looking at some of our really endangered mollusks. We've got good subfossil records. So Pupurangi kauri snails from Northland, and also these giant flax snails that I was talking about. So Pupu whakarango-tawa or Pupu harakiki, which are in all the sand dunes. So that photo at the bottom is one of the sand dunes up the top of Harangi Hill at Cape Maria Van Diemen. So we can really use these I suppose, fossil ecosystem to answer these really cool big picture questions around evolution and genetic diversity and bottlenecks and shape variation. So hopefully what I've shown you is by using paleontology and ancient DNA across the, I suppose, the breadth of New Zealand's geological history, is we need to look at the big animals, so the Brobdingnagians, as well as the small animals, the Lilliputians. And while species are having these individualistic responses, we are seeing commonalities that you're getting species like the wrens, say with northern southern lineages, the geckos, forest geckos, and northern southern lineages, and the forests doing exactly the same thing. So the beech tree example here, this is silver beech. There's been this big debate of whether forest at the height of the Ice Age retreated to the northern areas of both main islands and then expanded back out, or whether there was in situ survival and I suppose what you call cryptic microrefugia, like little patches and gullies that you see like up the Canterbury high country or central Otago. And this research is showing fairly unequivocally that you've actually got survival of silver beech and southern beech south of the central South Island beach gap in southern South Island and northern South Island throughout the Ice Age. So that's quite nicely, I know it conflicts with the pollen record, but it's quite nicely gelling with what we're seeing. The foresters doing this, and then we've got the animals that rely on the foresters showing a similar signal. So we can use the animals of proxy for fossils, but also for groups that don't fossilize that well. So happy to end there and answer any questions that you guys may have.